Funding for this program is made possible by the Corporation for Public Broadcasting, Louis B. and Louise Hirschfeld Coleman, Sue and Edgar Wackenheim III, and contributions to participating stations. Coming up, seeking a better way to combat the Zika virus.
We can try to find a chemical to hijack those receptors, and then we can cause a mosquito to be like, oh my God, yuck, stay away.
And studying zebrafish for clues to human development.
It's quite fascinating to look at embryos under a microscope because you can actually see single cells. You can see how organs form, whether the brain forms normally, whether the heart beats normally.
Exploring the frontiers of science. Probing cutting-edge technologies. Seeking answers to the big questions. Welcome to SciTech Central. The Zika virus is a public health crisis waiting to happen. As with many mosquito-borne diseases, spraying is the go-to prevention method. But some Florida scientists are doing research that could one day stop the disease in its tracks. Before 2015, few people had ever heard of the Zika virus. Then, news broke of an epidemic in Central and South America. In the vast majority of cases, infection is asymptomatic or brings mild flu-like symptoms. But in the unborn babies of pregnant women, it can cause a severe birth defect called microcephaly. In adults, infection occasionally leads to serious complications and even death. The Centers for Disease Control and Prevention estimates that by early 2017, nearly 1 million people in Puerto Rico will be infected. Cases have started showing up in Florida, and experts believe they could multiply rapidly. In Central Florida, efforts are underway to reduce the risk. The virus is spread by mosquitoes, so one way of fighting Zika is mosquito control.
Right here is almost subtropical. Right here is almost the same weather as in the Caribbean. The weather is warm almost all year long. The mosquito get a better chance to breathe right here faster than in the northern states.
Amador Rodriguez is searching for 2 species of non-native mosquitoes known to carry Zika.
We're looking for water sources. The water sources, that's where the mosquitoes gonna breathe. That's where the mosquitoes gonna lay the eggs. That's what they're gonna incubate and hatch. And the main concern is trying to get the people safe. The mosquito will lay the eggs there.
When our field inspectors are out looking for sources, they are looking for any little thing possible that can hold water enough to breed mosquitoes.
Inspectors collect mosquito larvae and bring them to this Orlando lab to be analyzed.
The mosquito female, after it's taken a blood meal, it develops its eggs and it can lay up to 200 eggs per batch. And those mosquito eggs have to dry out for 12 to 24 hours. And then once they get wet, they hatch into little larvae. And the larvae have 4 parts to their life cycle. It just takes about 5 days to go through that larval life cycle, and then they're off and flying.
Officials use data on where the larvae were collected to determine where to spray, being careful not to target harmless local species of mosquitoes that form an important part of the ecosystem.
They are pollinators, and they're part of the food chain. There's lots of things that eat them. As larvae, fish eat them, frogs eat them, some other aquatic organisms eat them. But as adults flying around, there's lots of birds that eat them, bats. There's quite a few things that eat them.
But some researchers believe sprays are ineffective against the 2 species that spread Zika. These mosquitoes don't swarm at night when spraying usually takes place.
Female mosquitoes are the only mosquitoes that bite.
Florida International University neurogeneticist Matthew DiGennaro regularly meets members of the public to answer questions about Zika.
I'm trying to get pregnant this year, not the best time, obviously.
He and other experts believe genetically modifying mosquitoes might be more effective than spraying.
One way is to release male mosquitoes which don't bite that will species specifically find the Aedes aegypti female and render her functionally sterile and unable to reproduce. And so we can achieve population reduction through this mechanism. And it has been done in the Caribbean, it's been tried in Brazil, and I think that It's a lot better than spraying, which affects so many different insects. What we need to focus on is reducing just the Aedes aegypti and Albopictus possibly as well, their populations.
Another approach is to modify the DNA of mosquitoes to react differently to smells, such as a human odor. The first step is to figure out which receptors in the mosquito's brain cause it to be attracted to humans.
And then we can try to find a chemical to hijack those receptors, and then we can cause a mosquito to be like, oh my God, yuck, stay away. So it would almost be like a designer perfume that is designed to annoy mosquitoes. We think that understanding how a repellent works will help us design the next generation of repellents.
But techniques like these are likely years from being practical. Meanwhile, officials on the ground know they have to try many options.
Especially with these two species, we cannot spray our way out of this because they're so elusive and they're hard to control their habitats, and there's little bits of water all over the place. When we do hear about risks in other areas, we're very quick to learn about things that are going on and what's working to control.
A changing climate leads to some surprising consequences. At Crater Lake National Park in Oregon, surface water temperature is on the rise, and an invasive crayfish is threatening the lake's native creatures.
Biologist Mark Bucktenica is scouring the shoreline of Crater Lake. Flying ants, lizards, and small toads are everywhere.
Aren't they cute?
But the critter he's looking for is much more elusive. Then his persistence pays off. This is the Mazama newt.
Mazama newt, found no place else in the world.
Crater Lake formed nearly 8,000 years ago after Mount Mazama erupted and the caldera began filling with rainwater.
We don't know when newts entered the caldera, but sometime thousands of years ago.
There were no fish or other predators in the newly formed lake, and the Mazama newt expanded and thrived.
It's playing dead on us now.
It was the undisputed top of the food chain, but not anymore. Because crawling beneath the surface of the lake, a new champion has emerged. The signal crayfish. Their story begins more than 100 years ago, back when getting to Crater Lake from Medford took 5 days by horse and wagon. To attract visitors, early conservationists began stocking the lake with game fish like trout and salmon. Craig Ackerman is park superintendent.
In the past, the national parks have done many things which people thought were good ideas at the time that turned out to be not so great ideas.
In 1915, park managers introduced the signal crayfish to feed those fish.
And that turned out to be a worse decision than stocking the fish in the first place, because the crayfish have become out of control.
And this is obvious out in the lake. Scientists at the park are finding that crayfish and the Mazama newts don't really get along.
We have a standoff. What's going to happen?
Not only do they compete for the same food, but studies done by park biologists show crayfish chase and harass the newts, causing them to flee.
They almost look like they're pack hunting a little.
And in some cases, much worse.
They're virtually the perfect invader. The Mazama newt, on the other hand, is an ideal prey.
After thousands of years evolving without predators, the newt lost its best weapon, a potent neurotoxin that can kill.
With the loss of its toxicity, it's left virtually defenseless.
But actually, crayfish in Crater Lake weren't thought to be such a problem until relatively recently. Surveying started in 2008, and scientists found newts had the advantage, occupying about half the shoreline. The crayfish had most of the rest. By 2014, the crayfish had taken over 75% of the shallows.
It's right here, Scott.
And that's not all, says biologist John Umick.
Got it. Big bags for this one. Oh my God. Crayfish are impacting all the organisms in the nearshore, not only newts. In crayfish areas, we don't find snails. You have to go outside of the crayfish-dominated areas to even find a snail or two. Instead of this great biodiversity area, it's down to 1 or 2 organisms, and that's it, besides crayfish.
Without these tiny organisms eating the algae, the crystal blue clarity of Crater Lake could be at risk. Crater Lake biologist Scott Gerdner suspects that climate change is playing a role.
4 in A.
The surface temperature of the lake has increased about 3 degrees in the past 10 to 15 years.
And it may just allow the crayfish to move faster. They just are more active at warmer waters. And it may allow them to have more success at reproduction so that their numbers have increased faster now.
The team surveys for crayfish each summer.
Surface. Oh my gosh. That's a lot of crayfish.
They set traps all along the shore and deep into the lake.
6.
They count, weigh, and measure their catch.
38.55.
And they tag and release some to find out how they travel.
Yep.
They've even found crayfish at 750 feet.
I'll be
Look at that.
It's amazing.
deepest known crayfish in any lake system. What are they doing at 250 meters?
Scientists expect that if crayfish continue to spread,
it's very possible that the Mazama newt would be eliminated from the lake if we didn't do anything.
But at this point, no one knows exactly what to do. Trapping, even intensive trapping, hasn't made a dent. Yet there are a few glimmers of hope for the Mazama newt. There's the possibility of building underwater barriers or fences to slow crayfish expansion. Another solution could be provided by the lake itself. Spring-fed pools like this emerge when avalanches pile up rock berms along the shore. These pools are newt nurseries.
They'll come into these pools while there's still snow on the edges of the pool.
The conservation promise is that while newts move across land to get to the isolated pools, signal crayfish do not. If other strategies to stop the spread of the crayfish aren't effective, these pools could become a final stronghold for the Mazama newt and other native animals.
We have the opportunity right now to at least slow down the invasion of crayfish. If we miss this opportunity, I think it's going to be a lot of trouble for the newts.
Without intervention, this unique creature could vanish from the lake within the next few decades. Controlling the signal crayfish and protecting this unique ecosystem will be labor-intensive and expensive.
But the Park Service's mission and mandate is to do this above all else. So we will put the resources into this that we feel necessary.
The sobering reality is crayfish will likely never be eliminated from Crater Lake.
Come on now, guy.
And maybe the best the national park can hope to do is carve out a few safe havens for the Mazama newt.
One fish in a UK lab tank did something so unexpected, it got a whole research paper dedicated to it. Scientists were keeping a bunch of cichlid fish in isolation, in separate tanks, as part of a study on species hybridization. And it seems that one of the female fish got bored or lonely or something, because apparently it fertilized its own eggs and had dozens of babies, all by itself. Researchers have called this solo style of sexual reproduction selfing. For now, researchers think that selfing might be a rare evolutionary strategy that some animals can use to reproduce in a new or changing environment. So thanks, nature. You're really weird.
The zebrafish is popular with aquarium owners everywhere. But scientists also love this small creature, not least because it can provide valuable insights in developmental biology.
The Stowers Institute for Medical Research in Kansas City has been described as cutting-edge technology, integrated with broad-based expertise to ask sophisticated questions. The research undertaken here falls in the category of basic or fundamental, which often leads to the question, what exactly does that mean?
I like to think of basic research as the engine for discovery. You need to make discoveries. And once those discoveries are made, they can serve as the basis or the foundation as a process so that those ideas could be translated, developed, and then eventually applied for human health or medicine.
And zebrafish are playing a role in that process in Tatiana Piotrowski's Stowers laboratory. These familiar-looking members of the minnow family are used as model organisms for studying developmental biology. Basically, how cells form tissues and tissues become organs. A zebrafish embryo grows outside its mother and remains transparent, so its developmental processes can be easily viewed.
It's quite fascinating to look at embryos under a microscope because you can actually see single cells move, interact with their environment. You can see how organs form, whether the brain forms normally, whether the heart beats normally.
Tatiana and her team are especially interested in investigating a sensory system called the lateral line that's found in all aquatic vertebrates. It's important to the schooling behavior of fish and allows them to detect prey or potential predators. The lateral line gets its name from the way multiple copies of its main sensory organ, the neuromast, are dotted along a fish's body and around the eyes.
And each sense organ possesses receptor cells, which are called hair cells, because they have cilia sticking out into the environment, which look like little hairs. And as water flows across these cilia, it actually sends a signal to the brain, and so that's how the animals measures the water movement.
The lateral line gradually develops from a group of around 100 cells called the primordium. The primordium first forms behind the fish's ear. It then begins migrating toward the tail tip. It's divided into the leading edge and the trailing edge.
It raises really a lot of interesting questions. So, for example, how does a group of cells know which direction to migrate? Do they just follow the cell in the tip, or does each cell know which direction to go?
There are several theories about how small cell clusters that look like rosettes eventually become neuromasts. And testing requires a whole lot of fish.
Adult zebrafish will lay up to 200 eggs per week. And this allows us to do large-scale genetic mutagenesis screens. What this means is we are disrupting gene function randomly with mutagens that we can apply on the adult fish. So we can disrupt genes, and then we look at the live embryo, and now you can see, okay, these cells are not behaving anymore like they were supposed to. And then this way, it gives us a tool to identify what these genes are normally doing.
Okay, so why is the Piotrowski lab focusing on a sensory organ in fish? Well, because neuromast hair cells, by which fish sense water movement, are actually very similar to those found in the human inner ear. However, our inner ear hair cells turn sound vibration into electrical activity, which nerve fibers transmit to our brain, allowing us to hear.
If you disrupt particular genes in the zebrafish lateral line hair cells, they stop functioning, and the same genes cause deafness in humans, for example.
A zebrafish's hair cells are just as susceptible as ours to damage and death, from overexposure to noise or the wear and tear of aging. But unlike us, they have a secret weapon. It's called regeneration, in which new hair cells regularly replace any that have been injured or killed.
Once we understand how regeneration happens in a fish, we will then be able to also compare it to a mouse and see where this process is blocked in a mouse or even in other animals such as humans.
The way I like to think about it is the model system is a way of learning how things are put together to understand cues and instructions, and then you can compare and contrast those. There are often instructions that are used primarily in a fish that also play a role in a human that we weren't aware of. But if you don't make the discoveries, you don't have a way of moving forward. Unfortunately, sometimes 90% of what we do doesn't work, but the 10% that do can shape the future of mankind.
Hello, how y'all doing? We're here today to donate aloe plants for all the students of Amy Scott's second grade class, as well as do a presentation.
I was very excited. We were always happy to have donations from community members, and the Collins brothers have been amazing to work with.
A big part of what we do, we talk about soil science. The plants actually have a process called phytoremediation, where they'll take toxins from the air and build their own tissue out of it. So anything that could be, say, coming from paint is going to disappear out of the air. And also it's going to give you oxygen, which is essential, especially for young children.
They think it's so cool to know that the plants in our classroom have been cleaning their air. I've noticed that my students have gained such a greater sense of responsibility. They take care of their plants, they water the plants on their own. Even their grades have improved a bit, and they love just having the plants in the classroom.
That we had grown off of a large mother at our house.
It's helping them learn and engage more in the classroom and just be more active students.
Just like your brain grows, the plant will grow if you read to it.
So the main focus is just trying to teach them all about nature and how it works, in hopes that we can take care of the planet. So if we can teach them how to properly take care of soil, we're going to have soil for generations to come. That's really the only way you're going to learn how to properly take care of a plant.
Plants are the foundation of every terrestrial ecosystem. But with rainfall declining in California, it's not clear how they'll adapt to this new arid climate. One researcher hopes to find out.
This is Jennifer Funk in her native environment, studying California plants. The Chapman University researcher wants to know which species in this field of flowers and coastal sagebrush can adapt to long-term drought conditions.
Is that enough leaf material on this individual leaf? Mm-hmm. Okay. Yeah.
It is forecast that Southern California will get less and less rainfall over the next century. Funk hopes her research will pinpoint which characteristics in plants help some survive a drought, while others perish.
So today we're here measuring photosynthetic rates. So it's a really good indicator of sort of the health or the functioning of the plant. And then in a couple weeks we'll come back out and we're going to dig what are called soil cores, where we actually dig a long tube of soil and we actually capture some of the plant roots in that. And from that we can look at things like root morphology and branching structure and some other characteristics about the roots.
The results could improve future environmental restoration efforts. Instead of replanting native species that may have been in an area 50 years ago, the study could identify different native plants that are better adapted to the drier environment. Funk's thirst for knowledge about what makes a plant drought-tolerant could have an impact around the world.
Well, Southern California has a Mediterranean-type climate, and we're actually only one of 5 Mediterranean regions across the globe. So it's quite possible that the stuff that we're learning here can be applied to different systems in South America, like in Chile or Western Australia, or even the Mediterranean basin.
Funk says there'll be winners and losers. We will lose some species that can't adjust to drought, while other plants will thrive. Those changes will also create a domino effect on the native wildlife.
So plants are really the base of the entire ecosystem, right? So they provide all of the energy through photosynthesis. They are what provide the foundation for all of the animals that live in that system. The type of vegetation community that you have 50 or 100 years from now is really going to impact the animal community as well.
Did you find anything out about F4? How many species there are in here?
So while this self-proclaimed plant nerd looks at these leaves and roots, she's really looking ahead to try and solve this growing problem.
That's all for this week on SciTech Central. Thanks for watching. Join us next week for more stories from the frontiers of science and technology. Funding for this program is made possible by the Corporation for Public Broadcasting, Louis B. and Louise Hirschfeld Coleman, Sue and Edgar Wackenheim III, and contributions to participating stations.